Hi, welcome back. So we were talking about ligand-gated channels, and as the name suggests, they work when the ligand binds to it, and more specifically, we can think of the voltage-gated channels. Okay, so cell surface receptors that bind to extracellular ligands are the ligand-gated channels, and their ability to conduct a flow of ions across the plasma membrane is regulated directly by ligand binding. So you have your plasma membrane here, this is my receptor, and I have this ligand binding to this receptor, and this will cause some ions, could be anything, sodium, potassium, whatever, depending on the membrane potential. So ions, it will allow some ions to flow through the plasma membrane, and this is actually directly regulated by the binding of the ligand. Now, flow of ions can result in temporary change in the membrane potential. Obviously, there will be a change in membrane potential, and membrane potential is just the difference in charge on the outside as compared to the inside of the plasma membrane, and this will affect the activity of other membrane proteins like the voltage-gated channel. So now, say if there's a lot of ions flowing in, then on the membrane you'll also have some voltage-gated channels, and now these voltage-gated channels, their activity will be affected too. Okay, so that's about ligand-gated channels. The next one is the steroid hormone receptors, and these are kind of cool too. Steroid hormone receptors. Okay, so what about these guys now? Well, they function as ligand-regulated transcription factors. Ligand-regulated transcription factors. So, I'll just try and draw a schematic here. So here's your plasma membrane. Here's your steroid. Steroid comes into the cell, and remember that their receptors are present in the cytoplasm. Okay, so steroid hormones diffuse across the plasma membrane and bind to their receptors, which are present in the cytoplasm. So this part is happening through diffusion. Okay, let me use a different color. And now there's a receptor inside the cell, and they bind to this receptor, so it is present in the cytoplasm. Let me write it: receptor present in the cytoplasm. Okay, so they get into the cytoplasm, they bind to their receptor, and this binding causes a conformational change. Okay, now, so I'm just gonna take my red color again, continue on with the schematic, and then I have my blue color. So now my receptor, my steroid hormone receptor, has bound to the steroid, and this causes a conformational change in this complex. So there's a conformational change, and now this conformational change allows this whole thing to enter the nucleus. So previously, just this receptor could not move to the nucleus. It needed that binding event to happen. So once that has happened, they move to the nucleus, and then they bind to elements present in the promoters or enhancers of hormone responsive genes. So you'll have your DNA or RNA or whatever, and say this is my promoter. So these things will bind here. So either they'll activate translation, transcription, or they can repress activation or translation, depending on the kind of signal. So this interaction gives rise to an increase or decrease in the rate of gene transcription. So they affect gene transcription, right? Just remember this word, ligand-regulated transcription factors, and the whole story will come to your head. Because as soon as you see transcription, you know what we're talking about. Okay? Several types of receptors act by unique mechanisms. Some, like the beta and the T cell receptors, that are involved in the response to foreign antigens, associate with known signaling molecules such as cytoplasmic protein tyrosine kinases. So that was just a little extra note. So the next topic is going to be G protein coupled receptors, background information on receptors and the pathway and whatnot. So we'll see you in the next video.